Hello everyone. Today we will know about the carbohydrate. So carbohydrate is called as the polyhydroxyaldehyde or polyhydroxyketone. So poly means many and the hydroxy means OH group. That means in the carbohydrate there are many hydroxyl group are present. That's why it is called as the polyhydroxy. And the aldehyde and the ketone, that means the carbohydrate can be present in the two form. One is in the aldehyde form, another is in the ketone form. That's why it is called as the polyhydroxyaldehyde or polyhydroxyketone. And again the carbohydrate is also called as the hydrates of the carbon. Hydrates means the formula of the carbohydrate is CnH2nOn. Look at this ratio H2nOn. This is exist in the same ratio as in the water. That's why it's called as the hydrates of the carbon. The carbohydrate is the most abundant biomolecules in the earth. That means any biomolecules, that means among protein, among nucleic acid, among lipid, in any biomolecules, carbohydrate is the most abundant biomolecules in the earth. Again, the carbohydrate can be divided into three parts: monosaccharide, oligosaccharide, polysaccharide. Monosaccharide means single sugar is present, is called as the monosaccharide. Oligo means 2 to 10 monosaccharide, they are joined together to form the oligosaccharide. And the polysaccharide, it is when the more than 10 monosaccharide they are joined together, then it is called as the polysaccharide. Please note that this polysaccharide is also called as the non-sugar because it is not sweet in the taste. That's why it is called as the non-sugar. Now come to the monosaccharide. In this slide we will know about the monosaccharide. So monosaccharide, the formula is the CnH2nOn. So Cn and 2 times of the H and same times of the oxygen. So in the monosaccharide, minimum carbon amount should be 3 and maximum is 7. That means if in this formula, if minimum amount of the carbon is C3, then C3H6O6. It is called as the triose because of the presence of 3 carbon. Then if the carbon amount is 4, then it is called as the tetrose. Then if it is 5, then it is called pentose, then hexose. Then if it is in 7, then it is called as the heptose. So if you will go about the properties of the monosaccharide, then it is the colorless, it is crystalline solid, it is soluble in water but insoluble in non-polar solvent. So now come to the chemistry part. The monosaccharide can be exist in the aldehyde form or in the ketone form. Let's say in the triose, that means C3H6O3. Both these structure have the same formula C3H6O3. Here is also C3H6O3, but it is found in the two different form. That means in one form it is the aldehyde form and another in it is the ketone form. So the carbohydrate are in the triose. If it is present in the carbohydrate form or the aldehyde form, then it is called as the glyceraldehyde. If it is present in the ketone form, then it is called as the dihydroxyacetone. Like that, this is the C5 or it is called as the pentose. The ribose, ribulose, they both have the same formula C5H10O5, but they exist in the two different form. One in the aldehyde form, this one, and another is in the ketone form, this one. So the aldehyde form is called as the ribose, the ketone form is called as the ribulose. So like that, it is the hexose. That means C6H12O6. They both have the same formula, but in both different structure. The aldehyde form is called as the glucose. The ketone form is called as the fructose. Please note that this fructose is also called as the fruit sugar, because the fructose is present in the naturally in all the fruits. That's why it is called as the fruit sugar. Okay. Now come to, there are different types of the monosaccharides. For example, this is the glyceraldehyde. This is the C3. Then this is the C4. That means erythrose, threose, they are the C4. And these are the C5. That means ribose, arabinose, xylose, lyxose, they are the C5. And the C6, they are the allose, maltose, glucose, mannose, glucose, idose, galactose, talose. So they all are the formed by this glyceraldehyde, but this is only in the aldehyde form. In the ketone form also there are different types of the monosaccharides, and they are formed by the this dihydroxyacetone. Okay. So now come to the enantiomer. What are the enantiomer? There are the two types of the enantiomer. One is the D, another is the L. D is the dextrorotatory, L is the levorotatory. So what is the dextrorotatory? Dextrorotatory means any molecule which can rotate the plane of the polarized light to the right side is called as the dextro. And the levo means any molecule which can rotate the plane of the polarized light to the left side is called as the levorotatory. And they are the mirror image. That means if a mirror is introduced between them, then this structure reflects in the mirror. So now, how do you know that whether it is the D or the L? By this structure. Now see this structure or the functional group, that is the CHO group or the aldehyde group. Let's say in this group, this is the carbon which is called as the chiral carbon. What is the chiral carbon? The chiral carbon is the carbon to which 4 different groups are joined together. That means in this carbon, one is the CHO, another is the hydrogen, another is the OH, another is the all total groups are joined together. So 4 different groups are joined together. So now this is called as the chiral carbon. I can come to this carbon. To this carbon, one is one group, another is another group, another is the hydrogen, another is this OH. So now again 4 different groups are joined together. So this is also called as the chiral carbon. But now comes to this carbon. To this carbon, 2 hydrogen groups are joined together. So because of the same groups are joined together to the same carbon group, that's why this carbon is called as the achiral groups. So if in exam it will be asked that, let's say this is the hexose, that means 6 carbon is present. So if in the exam it will be asked that in the hexose how many chiral carbons are present. So always it is n minus 1. That means if it is hexose, the chiral carbon is 5. If it is pentose, that means 5 carbon, the chiral carbon will be n minus 1, that means 5 minus 1, it is 4. Okay. So now come to again to the enantiomer. What is the enantiomer? It is the dextrorotatory, another is the levorotatory. So now how do you know whether it is the dextro or the levorotatory by the structure. Now this is called as the functional group, that is the aldehyde group, and this is the chiral carbon, and this is the farthest chiral carbon from the functional group. That means the functional group is the aldehyde and this is the farthest chiral group. This is because this is not a chiral carbon and this one is the farthest chiral carbon. So in this chiral carbon always one hydrogen, another is the OH group is attached. If the OH group is attached to the right side, then it is called as the dextrorotatory. If the OH group is attached to the left side, it is called as the L or the levorotatory. That means always the farthest chiral carbon from the functional group. If to this carbon the OH group is present at the right side, it is called as the dextrorotatory. If it is present at the left side, then it is called as the levorotatory. Now in this D-glucose, because the chiral carbon is this and here the OH group is attached to the right side, that's why it is called as the dextro. If it is present at the left side, it is called as the levo. That's why in the D-mannose, D-glucose, D-galactose, because in all these structures the OH group is attached to the right side. So this is called as the enantiomer. Now called as the epimer. Epimer is the any two structure which are differed by single carbon. That means let's say this is the mannose, this is the glucose. In these two structure only differs only by this carbon. This is the C2, C1, this is the C2. In the C2 only the OH and the H is reversed. That means in the mannose OH is present at the left side, but in the glucose OH is present at the right side, and here it is present H in the right side, but here it is the H present in the left side. So only the mannose and the glucose, they are differed only by at the C2 primer. That means only differs by the one carbon. So mannose and the glucose are the epimers. Like that, if you will see in the glucose and this is the galactose, here they differ only by this carbon 4 group. That means in the carbon 4 group, the OH and the H are the reversed. So they are the glucose and the galactose, they are epimers at the C4. But note that the mannose and the galactose, they are not epimers because they are differed by the C2 and C4. That means there are two different carbons. So that's why the mannose, galactose, they are not epimers each other. But the mannose and the glucose, they are the epimers to each other, while the glucose and the galactose, they are also epimers to the each other. Okay, now to the enantiomer. This is the L-glucose, then another is the D-glucose. Let's come to this structure. This is the C3H6O6, O3 or triose. They can be present in the two forms. One is the glyceraldehyde, another is the dihydroxyacetone. Look at this dihydroxyacetone structure. In this dihydroxyacetone structure, there are no chiral carbon. That means this carbon is not chiral because two hydrogen groups are joined. This carbon is not chiral because it is joined to one CH2OH and again same CH2OH group is attached. And this is again not chiral carbon because again two hydrogen groups are joined or two same molecule is joined. So in the dihydroxyacetone there is no chiral carbon is present. So that's why it cannot rotate the plane of the polarized light, because which can rotate the plane of the polarized light? At least it should have one chiral carbon. But this dihydroxyacetone, that means in all the carbohydrate, the dihydroxyacetone, it is the only exception which has no chiral carbon. That's why the dihydroxyacetone cannot rotate the plane of the polarized light out of the all the carbohydrates. Now again come to the cyclic form of the sugar. That means when the monosaccharide, they exist in the aqueous solution, they are formed in the cyclic manner. That means this is the extended manner, but they can also exist in the cyclic manner in the aqueous solution. How? Now they when the C5OH group attacks at the C1, then it looks like this, or it can be seen by this structure. This is the 6-ring structure. 1, 2, 3, 4, 5 and 6. This is the 6-ring structure. So in the aqueous solution, it can exist in the 6-ring structure, which is called as the pyranose. Again, this structure, it can exist again by the 5-ring structure, because look at this structure. This is the same structure, but now the C5OH reacts with C2 of the carbon. Now it forms the C5 or the 5-ring structure, which is called as the furanose. So if exists in the 6-ring structure, it is called as the pyranose. If it exists in the 5-ring structure, it is called as the furanose. So pyranose in this structure, this is called as the carbon 1, this is 2, this is 3, this is 4, this is 5, and this is the 6th carbon. And in the 5, it is 1, and this is the 2, this is 3, this is 4, this is 5, and this is the 6th carbon. So the monosaccharide can exist in the two different forms, either in the 6-ring structure or in the 5-ring structure. 6-ring structure is called as the pyranose, 5-ring structure is called as the furanose. Actually the chemistry is the aldehyde group. If it reacts with the alcohol, then it is called as the hemiacetal, like this. This is the hemiacetal form. Again, the ketone, if it binds to the alcohol, then it creates the hemiketal. So this is the hemiketal form. So always the pyranose is the, most of the pyranose is the hemiacetal and furanose is the hemiketal. So now how the pyranose and the furanose name comes? In the chemistry, like in the benzene, in the chemistry there is a structure, it is called as the pyran, and another structure is called as the furan. The pyran looks like the 6-ring structure, that's why it is called as the pyranose. And the furan looks like the 5-ring structure, that's why it is called as the furanose. Okay. Now comes to the anomere. Anomere means there is an anomeric carbon. Anomeric carbon means always the anomeric carbon always at the 1 or the second, first or the second carbon. That means if in the maltose, let's say maltose is a disaccharide and it is formed by 2 glucose. That means it is a glucose, another glucose is joined. So it joined to the at this place, at the C1 place. So that's why it is called the anomeric. But let's say in the fructose, let's say the sucrose. Sucrose means one glucose, another is the fructose. It is called as the sucrose. So in the sucrose, it is joined to at this condition, and this is the second carbon. The first carbon is that. So always in the fructose, the anomeric carbon is the second carbon, but in the glucose, the anomeric carbon is the first carbon. So what is the anomere? Again, this is the glucose extended form. It can be exist in the two form. One is the 6 ring structure or in the 5 ring structure. In the 6 ring structure, this is the 6 ring structure. One is called as the alpha glucose, or it's called as the alpha glucopyranose because it is present as the 6 ring structure. If it is the glucofuranose, that means same glucose present in the 5 ring structure. So now it is called as the alpha glucopyranose, and another is called as the beta glucopyranose. So what is the alpha and another is the beta? So at the anomeric carbon, if this hydroxyl group, if it is present at the lower side, this is called as the lower side. One is the upper side at the hydrogen, lower at the OH group. If the OH group is present at the lower side, it is called as the alpha. And again, if the OH group is present at the upper side, it is called as the beta. In this structure also, the OH group is present at the upper side, that's why it is called as the beta fructose. If it is present at the lower side, it is called as the alpha fructose. Okay. Now comes to the derivatives of the monosaccharide. That means monosaccharide can be modified to form the different types of the structure. For example, one is called as the glycoside. What is the glycoside? Now remember this structure. This is called as the aldehyde. Another is the alcohol. Aldehyde reacts with the alcohol to form the hemiacetal. Now in the glycoside, the aldehyde forms to the alcohol is called as the hemiacetal, and again if the hemiacetal again reacts with the alcohol, then it is called the acetal, and then it is called as the glycoside. So if the carbohydrate is the glucose, it is called as the glucoside. The suffix side is called as the glycoside. That means if the carbohydrate is glucose, then it is called as the glucoside. If it is the galactose, it is called as the galactoside. One of the example of the glycoside which will be asked in the exam is OABN. It prevents the sodium potassium pump. You know, in the, let's say in the plasma membrane, there is a pump. It's called as the sodium potassium pump. That means it allows the transport of the sodium potassium pump, which is active transport. You will study in the active transport of the plasma membrane. So in the OABN, this is a glycoside. It prevents the sodium potassium pump. Another example of the glycoside is streptomycin. You know, obviously it is a antibiotics. So now another is a sugar acid. Sugar acid means, this is the glucose. Let's say this is the functional group, that is the aldehyde. If the aldehyde group is oxidized to form the COOH group, then it is called as the aldonic group or aldonic acid. It is called as the gluconic acid because here the carbohydrate is glucose. If the glucose, it is called as the gluconic acid. If it is galactose, it is called as the galactonic acid, like that. If both means this group and the terminal group both are oxidized to form the COOH, it is called as the aldaric group. Same, if it is glucose, then it is called as the glucaric acid. If it is galactose, then it is called as the galactaric acid. So if only the terminal group is oxidized to form the COOH group, it is called as the uronic acid. If it is glucose, it is called as the glucuronic acid. If it is galactose, it is called as the galactonic acid. Okay, so these are the sugar acids. Another is called as the sugar alcohol. Sugar alcohol means this is the aldehyde group and here it is the CO group. That means the carbonyl group. The CO group, if it is reduced to form another OH group is formed. So like that, in the glucose, another OH is joined. So the glucose, if the terminal aldehyde group or the carbonyl group of the aldehyde group is reduced to form again OH group is added, then it is called as the sugar alcohol. For example, the glucose. The glucose, it is from the sorbitol. It is the xylitol. Xylitol is the, remember one structure. Now this is called as the xylose, is the C5. So now this is the xylitol, it is comes from the xylose, and here the suffix is the etol. That means glucose is the exception. That means if it is the glucose, it is called as the sorbitol, but if it is the xylose, it is called as the xylitol. Another is the another example of the sugar acid is the ascorbic acid or the vitamin C, which prevents the scurvy. So in the exam it will be asked that the ascorbic acid is the which type of the carbohydrate? Always answer will be the sugar acid. Another is the amino sugar. So amino sugar means this is called as the glucosamine. That means let's say in glucose. In the glucose here a OH group is present actually. But here in the glucosamine, NH2 group or the amine group is replaced. So that's why it is called the glucosamine. Means in one amine group is attached. So this is called as the glucosamine group. But sometimes the glucosamine group and to this amine group one COCH3 group is added. That is called as the acetyl group. That means another acetyl group is attached. So now it is called as the acetyl glucosamine. So or simply short form is called as the N-acetyl glucosamine or NAG. You know the NAG, another is the NAM or the N-acetyl muramic acid. In the cell wall of the bacteria, it is joined together by NAG and the NAM, or in the chitin also. It is joined by the NAG and the NAM. That means N-acetyl glucosamine. Again, it is joined to the N-acetyl muramic acid, which is another the amino sugar. So you will study at the polysaccharides. So these are the amino sugar. So these are the different types of the modified of the monosaccharide. And in the next slide, we will study about the disaccharide and the polysaccharide. If you like the video, please like and subscribe the channel. Goodbye.